All right, my friends. Welcome back. Happy Monday to you. This is an interesting week because it's the week of your spring day. Woo! Right. Friday, we won't have class. I'm sorry about daylight savings. I really am. It's murder on the sleep schedule. So, nevertheless, today's going to be kind of a tough lecture because what we're going to be talking about is, for part of it, is the synthesis of cholesterol. There's a lot going on there. There's a lot of stuff to draw, but I'm hoping to show you how everything that happens is an example of reactions that you've seen before, and it's going to involve carbocation stability, and it's going to involve alkenes acting as nucleophiles. It's also going to involve a little bit of epoxide chemistry. So we're going to learn the early biosynthetic pathways towards cholesterol. And the reason we're going to do that is because cholesterol is an essential molecule, not only it's a major component of cell membranes and can affect their properties, but also it's the precursor to lots of different hormones and vitamins and bile salts. So a lot of useful things come out of cholesterol. All right, so what I've shown you here is the molecule geranyl pyrophosphate reacting with a molecule of isopentenyl pyrophosphate to give you a 3-carbon. I'm sorry, I can't stand that color orange. That's kind of gross. All right, let's try again. That's a little better. 2 isoprene units in geranyl pyrophosphate, and then we're going to bring the 3rd isoprene unit in isopentenyl pyrophosphate. The mechanism here is going to be one that we discussed last time. The leaving group of geranyl pyrophosphate will leave to give a resonance-stabilized carbocation, and then the pi bond of isopentenyl pyrophosphate will attack that carbocation at the primary carbon to give you a new bond between carbon 5 of the orange unit and carbon 1 of the yellow unit, and then there'll be a subsequent elimination reaction that puts this double bond in the right place, and this molecule is called farnesyl pyrophosphate. Okay, so drawing that mechanism should be straightforward. What happens next in cholesterol synthesis is, well, if you count the number of carbons in cholesterol, there's 30, whereas there's only 15 carbons in farnesyl pyrophosphate. So we need to combine 2 molecules of farnesyl pyrophosphate together. Now, in principle, you could imagine to get to 30 carbons, you just tack on 3 additional isoprene units via this conventional approach where with each bond that we form, carbon 5 of one subunit is attached to carbon 1 of the other. And this kind of bonding we're sort of going to call, I've talked about the dimethyl tail of these isoprenes and terpenes. You've got the tail and you've got the sort of leaving group head. And all of these units are connected in the same orientation, head of one isoprene unit to the head of one isoprene unit to the tail of another. But it turns out that to get the molecule cholesterol, we actually need to link 2 of these farnesyl pyrophosphate groups together head to head. That is, we need carbon 1 of one farnesyl pyrophosphate linked to carbon 1 of the next farnesyl pyrophosphate. So to illustrate this, I'm going to use some different colors for, the isoprene units here. And so you have an enzyme called Squalene Synthase, which takes 2 molecules of pyrophosphate, farnesyl pyrophosphate, and links them together to form a 30-carbon molecule called squalene. And in the process, we're going to replace the bond between carbon 1 and the leaving group on each molecule of farnesyl pyrophosphate with a single carbon-carbon bond between green carbon 1 and yellow carbon 1. And that's going to involve a reduction reaction, going from something that used to have a carbon-oxygen bond to something that has carbon-carbon bond. That's a reduction. And so this is going to involve NADPH. Biochemists often list plus proton there to indicate that you need a proton transfer. We know that there's no such thing as a proton floating around on its own, at least not in aqueous solution, but we're going to tolerate that description of things. You'll learn more about this, and we're not going to actually talk about this step at all. The mechanism is known, and I talk about it in my grad level organic chemistry course, but probably we can just accept that it happens. If you want to know the mechanism, tell me you're coming and I will take a few minutes to review it myself, and then I can show it to you because it's complicated enough that I need to check. Anyway, so when we do this, we're going to take this molecule, let's see, 3 isoprene unit starting material, and we're going to create a new bond between carbon 1 of the yellow isoprene unit and carbon 1 of the green isoprene unit. 1, 2, 3, 4, 5. Okay, so there's the green isoprene unit, and let's see, 1, 2, hold on. Did I, Joshua, how long are you guys going to let me do that, huh? I numbered things wrong. Not over here, that was numbered right, but over here I numbered things wrong. So, we're going to take carbon 5 of the yellow isoprene unit, and we're going to link it to carbon 5 of the green isoprene unit. So for those playing along at home, carbon 1 is at the tail, carbon 5 is at the head. Sorry about that. 5, 4, 3, 2, 1. And then we'll draw the next isoprene unit. 5, 4, 3, 2, 1. 5, 4, 3, 2, 1. Okay. And gotta get the right colors. Okay, so that's a 30-carbon molecule called squalene. And now we've got all the carbons in place to form cholesterol, to form the core structure of cholesterol. Now, if you've heard anything about steroids before, you have 3 fused 6-membered rings and a fused 5-membered ring. Sometimes people call this 3 bedrooms and a bath. I don't see it, but if you want to remember it that way, you can. But you've got a very complicated structure that you're trying to get from a linear precursor. But what I want to show you is how the connections here, the positions of the methyl groups and of the alkenes relative to each other, sets this molecule up perfectly for a cascade of reactions that will make all of these rings in what is basically a single step, though we're going to take it a little bit slower than that. So nature will first use an enzyme called Squalene Monooxygenase. Do you need to know that enzyme? No. But it takes molecular oxygen, and FAD is a cofactor, and it makes an epoxide at the purple, it converts the alkene in the purple isoprene unit to an epoxide with this stereochemistry. Okay, so this is sometimes called squalene, just squalene epoxide, though sometimes people call it something different. And you know from 351 that epoxides are electrophiles. So, and you've also been used to, over the past couple of days as we've been talking about terpene chemistry, thinking of alkenes as nucleophiles. So what I'm going to do now, I think, is maybe confusing. I'm going to redraw this molecule in a shape that's going to make it a little bit clearer as to how we form a six-membered ring, okay? So draw it along with me, and then as we move on, I'm going to often copy and paste things because we won't have time to keep redrawing this molecule, so I hope that's okay. And even if it's not, I'm still going to do it. Okay. So this is the purple isoprene unit. And here is the epoxide in front. I guess I could draw this bond a little bit better going back into the page. That purple isoprene unit is connected to the orange isoprene unit like this. 1, 2, 3, 4, 5 with a double bond there. That's the orange isoprene unit. You can see that carbon 2 of purple and carbon 4 of orange are close to each other. And we've got another bond between isoprene units, and then 1, 2, 3, 4, 5. That's going to be the yellow isoprene unit. Then we've got the bond, the head-to-head bond between carbons 5 of yellow and green respectively. So that's going to be this bond here. And then we're going to draw the green isoprene unit, 5, 4, 3, 2, 1. And maybe I need to redraw this to have, so that we can tell that this stuff out here is in front. This is the green isoprene unit, and there's going to be a pi bond here. And then another bond between isoprene units. What we're going to draw now is the blue one, 5, 4, 3, 2, 1. Sorry, that's kind of ugly. Let me try again. 5, 4, 3, 2, 1. And then 5, 4, 3, 2, 1. I think that's right. Nope. Sorry. And this is the bond between isoprene units. 5, 4, 3, 2, 1. Okay. So I'm sorry about that. I know that took a while, but I think that labeling these things and showing the molecule in this perspective is going to help you see what happens. Your text is going to take you through this process, but rather than draw it like this, you're just going to draw it from above, and I think that can be somewhat confusing. So hopefully this is going to show you how we get the molecule. So what we've basically done is we've picked up this group and turned it around in front of the page, and that's why the epoxide's now facing us. So what do you remember about epoxides from 351? If you've got a strong nucleophile and an epoxide, and if there's a choice between two carbons, which carbon does the strong nucleophile attack? Less substituted one because sterics, but if the epoxide is first protonated, then which carbon does the nucleophile attack? More substituted, good. And that is because the epoxide begins to leave and the bond between oxygen and the more substituted carbon gets longer and weaker. And then the more substituted carbon experiences a partial positive charge. And that's why the OH group starts to leave from that carbon, because the more substituted carbon can better bear the partial positive charge. Okay, so epoxide chemistry that you've already learned about tells you that the reactive portion of this epoxide is right here on carbon 2. Notice that 1, 2, 3, 4, 5, 6 atoms away is an atom that's involved in an alkene, and you know that alkenes can be nucleophiles, right? So we're going to have this alkene act as a nucleophile to attack the epoxide. Now, as the pi bond attacks the epoxide, we're going to make a new sigma bond, and we're going to have to make it either with orange 4 or orange 2. One's going to get the sigma bond, one's going to get a positive charge. Based on what you know, who should get the positive charge? 2, right? Because it's more substituted, because more substituted carbocations are more stable. So here's the new bond we form, and that opens up the epoxide. Okay, now I'm going to show you what happens in this process step by step, though there is evidence that what I'm going to show you all happens in the same basic step. And we'll talk about that a little bit later, but we're going to go through it step by step because it's simpler that way. So I'm going to highlight in blue, this weird color of blue, the new bond that we made. And I'm going to redraw these methyl groups to make this look like a better chair. Okay, we just made a 6-membered ring, and we now have a positive charge on carbon 2, orange 2. But notice that we have, again, 1, 2, 3, 4, 5, 6 atoms away, we have yellow 4 involved in an alkene, and if an alkene can act as a nucleophile to open an epoxide, surely it can act as a nucleophile to attack a carbocation, right? So we'll draw an arrow from the pi bond between yellow 2 and yellow 4 to make a new bond there. Okay, and I'm copying and pasting. Don't worry about that. If you're writing on paper, all of this stuff's going to be in the notes, and it's all also shown in your book. If you feel like it would be useful, you can come back later and draw the individual steps. Here is the new bond we just made. I'm going to rearrange that methyl group so that you can see more clearly that it would be up. And again, we're going to have a positive charge on the more substituted carbon. So in making these 2 bonds in blue, so far, we followed the Markovnikov principle, which is that when you add to alkenes, you add first to the less substituted side, so the more substituted side can have the carbocation in the intermediate. Okay. Now, next step, we've got to the point where the yellow isoprene unit is now bonded head to head with the green isoprene unit. And this changes things somewhat because it puts carbon 2 of the green unit rather than carbon 4 close to the positive charge. Now, if we formed a bond between green 2 and yellow 2, that would be an anti-Markovnikov addition because it would leave a positive charge on this less substituted carbon. So you could consider an alternative. What if instead we had carbon 4 attack, green 4 attack yellow 2 to leave a positive charge on green 2? That would be a 1, 2, 3, 4, 5-membered ring. but it would give us the more substituted carbon. So you've sort of got a trade-off here. You can get the more substituted carbocation, but you have to make the more sterically hindered 5-membered ring, or the more strained 5-membered ring. Or you can make the more stable 6-membered ring, but have to get the less stable carbocation. And so what I'm going to show you now is what actually happens. As to why it happens this way, it's either that what I'm going to show you actually is the more stable option, all things considered, or it's that the enzyme guides the process to have this particular outcome. There's a computational chemist who visited our department several years ago who's done detailed calculations on some of these intermediates, and his calculations show that the pathway that we're showing you here is actually the lowest energy pathway. So for what it's worth, we can show, oops, sorry, that's, need to copy and paste again. So what will happen is that the pi bond between 2 and 4 will attack yellow 2, and we're going to make a new bond here between the 2 carbons, and that's going to leave a positive charge on green carbon 4. And again, that's the less stable carbocation, but it gives you the more stable ring. Except now we're going to turn around and do the opposite. We're going to have the pi bond here attack carbon 4 to make a new bond between green 4 and blue 4. And the reason, and when we do that, that's going to give us, oops, that's not what I wanted. That's going to give us a 5-membered ring here with a positive charge on the more stable blue 2. So here are the 4 carbon-carbon bonds we just made. And I got to get rid of that positive charge, sorry. And I also need to draw this bond better. Okay, questions so far? 4 bonds. The first 2 follow the rules that we've learned so far. The last 2, you have a trade-off between forming a 6-membered ring versus a 5-membered ring, and a secondary carbocation versus a tertiary carbocation. In the first case, you go for the 6-membered ring but secondary carbocation. In the second case, you go for the 5-membered ring but tertiary carbocation. Why is it that way? I don't know. Would I expect you to predict that? No. But if you know what happens, could you draw the appropriate arrows based on what you know is chemically reasonable? Yes. All right. Or would I expect you to be able to do that? Yes. Okay, so I'm going to draw this now from above to give you a sense for what we're looking at. Okay, so you're going to have an OH group here, 2 methyl groups there. Remember how I told you can recognize a terpene by looking for the dimethyl tail? There it is, right there. Here's a bond between isoprene units. Here's the next one with a branching methyl group. Then there's a bond between isoprene units, and then here's the next one, again with a branching methyl group. Oops, wrong way. This is a bond we made during the cyclization process, as is that one. This is the head-to-head bond between yellow isoprene unit and green. Let's see, what else have I missed? 5, 4, sorry, there's a up methyl group here. Then there's a bond between isoprene units. Five, 4. And then, I'm running out of space. Lastly, the pink isoprene unit, 5, 4, 3, 2, 1. Okay, so that's sort of what we've done. And though I've shown it to you step by step so you could see each individual bond forming, and by the way, sorry, there's still a positive charge here. What your text will do, and what people generally show when they're drawing the mechanism of cholesterol formation, is all of these arrows happening at once. That is, instead of drawing a carbocationic intermediate at this stage, they show the pi bond attacking just exactly as that carbocation is forming. And then we're going to do the same thing here. Just as soon as the carbocation here is forming, we'll have the pi bond attack. And again, just as soon as this carbocation is forming, we'll have this pi bond attack. So your text will show 1, 2, 3, 4, 5 arrows all at once, stitching together 4 rings in one single step. And there's evidence that there aren't really carbocationic intermediates along this pathway. But it also seems unrealistic that all of these things should happen simultaneously. So what we're likely seeing is something in the continuum between a single-step reaction like that versus a multi-step reaction like this, where there are discernible intermediates and transition states. What if instead of that we just had sort of a very broad transition state without discernible intermediates? It's sort of flat, but it actually takes a long time. That's probably what's going on here, at least according to the calculations. So it's sort of a rugged potential energy landscape with a plateau rather than a peak. And that's at least what the evidence suggests is happening. But I find it amazing that you can take this simple linear precursor, make it an epoxide, and then all of the sudden protonate the epoxide, and boom, these bonds stitch together in just that way. And again, the enzyme guides this process and gives you the correct stereochemistry, but the pathway that is involved in these cyclizations turns out to be the lowest energy pathway. All right, questions so far? We're not done, unfortunately, in our synthesis of cholesterol, but we're getting there. Go ahead.
Did you say there were 4 bonds formed?
4 bonds formed. 1 here, carbon-carbon, 2, and 3. Oh, I'm missing a blue. Sorry, this one.
Oh, okay.
Yep.
Okay.
Yeah, thanks. That's all right. Okay, other questions? Okay. I'm going to show you, and I'm missing also a methyl group, aren't I?
I do have another question.
Okay, go ahead.
Why doesn't it do the last stitch? There's a carbocation and there's an alkene.
Yes, why couldn't you just cyclize here? So let's count where the carbocation is versus where the double bond is. 1, 2, 3, 4. So you could form a 4-membered ring and get a positive charge here. Or you could form a 5-membered ring and get a positive charge there. I don't know why that doesn't happen. It doesn't, though. You don't observe the last possibility there. And part of it likely has to do with all of the uses for cholesterol. So cholesterol itself is going to continue to have this group there, which is sort of going to be a nonpolar tail. And cholesterol is still going to have this OH group. And so that's going to give it the ability to participate in lipid membranes because it's sort of got a polar head group and a nonpolar tail. But in the case of a lot of different steroid hormones, they're actually going to cleave off a lot of this and put different functional groups there. If you go and look at the difference between estradiol, progesterone, and testosterone, a lot of it's out here. And so I think there's a need for some chemical diversity there that somehow precludes having a ring. Really, that's an evolutionary question. Why does biology need it to be that way? And I've got nothing, so sorry. What else? Okay, well, just when you thought things couldn't get worse, they're going to, because what happens next is a bunch of carbocation rearrangements, which I know you hate. Now, in 351, you were taught that you can have a carbocation rearrangement whenever you have, say, a secondary carbocation adjacent to a tertiary carbon, because then, via what you learned was a 1,2 hydride shift, you can convert a less stable secondary carbocation into a more stable tertiary carbocation. That look familiar? That's called a 1,2 hydride shift. And then you were also told that if you have a secondary carbocation adjacent to a quaternary carbon, that actually you can do a 1,2 methyl or alkyl shift, wherein the methyl group leans over, and that converts a less stable secondary carbocation into a more stable tertiary carbocation. That's a 1,2 methyl shift. What I think, at least when I teach 351, I tell students, I'm not going to do this to you because doing so would be mean. Nevertheless, you may not have been, I guess by analogy to what's above, we ought to put this here. You probably didn't spend a lot of time thinking about carbocation rearrangements that would allow a tertiary carbon to rearrange to a tertiary carbocation to rearrange to another tertiary carbocation. Nevertheless, that should be reasonable because you're just trading one tertiary carbocation for another. Now, you could imagine a lot of things happening with this positive charge, but the intermediate that is the goal is going to end up having a double bond between these two carbons in the yellow subunit. And so we've got to rearrange the groups to get this carbocation all the way here. And so what we're going to see is a cascade of 1,2 carbocation rearrangements. I'm not going to draw the intermediates because there's evidence that this happens all at once, and I think conceptually you can get the idea of a 1,2 hydride shift that puts the proton there and the positive charge there. You got that? Sort of can see that? Should we draw the intermediates? We can. It's just going to be a lot of copying and pasting. Okay. Then, as there builds up a positive charge here, you've got an adjacent carbon-hydrogen bond that's going to lean over, do a 1,2 hydride shift, and now this green carbon-4 is going to have the positive charge. Then you have this methyl group that's going to lean over and do a 1,2 methyl shift, and that's going to put a methyl group on this carbon and leave this carbon with a positive charge. And then this last methyl group is going to lean over and do a 1,2 methyl shift, which will finally put the carbocation where we want it. I don't know why we want it there. That's a great question to ask. And it may have to do with stability of intermediates. It may be that having the positive charge on the carbon I'm going to show you is better than having it somewhere else. Also, moving these hydrogens, many of them are at carbons that are shared between two rings. This can change the conformation of the rings and might lead to a more stable arrangement. I haven't thought through that or done that analysis at any level of detail, so it's possible that's just hand waving. But one of the interesting features of this rearrangement process is stereochemistry is conserved. That is, this proton, which was down, is still going to be down on the other carbon because it leaned over on the same face of the molecule. The methyl group, which was up here, is still going to be up there. The methyl group, which was down here, is still going to be down there. So this may change the stereochemistry of the ring, and it may get you to a more stable conformation or a more desired shape. I don't really know. Now you have a positive charge here, and it's at this stage that a base will come along and remove this proton, and that will get you to a neutral product. Nope, don't convert that to text. I'd like to see it try. Yeah, we got to actually try that now. Convert to text. Interesting. T with 4 divisions. Yeah, that's inspiring, isn't it? Got to work on that AI. It's like Siri saying, I'm sorry, I didn't understand that. So now you have a double bond there between these 2 rings. This is not yet cholesterol. This is called Linosterol, and it is the precursor. There are actually, get this, 19 more steps between this and cholesterol. And the steps involve oxidizing and moving around methyl groups, getting rid of some of them. These 2 disappear. And then once you got to cholesterol, then you can convert that into hormones. You can convert it in, I guess cortisol would be included in the hormones. So you've got progesterone, estrogen, you've got testosterone, cortisol, and then you also have vitamins like vitamin D, and then you also have bile salts, which homogenize lipids and fats in your diet. And so all of these things come from cholesterol. But the core ring structure stays the same from this point out. All right, other questions?
Just to clarify, because the 1,2 hydride method shifts,
Uh-huh.
It starts out as below, it will shift and stay below.
That's right. Stereochemistry is conserved. So if the hydrogen was down here, and this sort of makes sense because if the hydrogen's going to lean over and make a new bond, it probably isn't going to lean over like this. Such things can happen, but not usually with 1,2 rearrangements. Okay, what else? All right, so I know the question on all of your minds is this going to be on the test? And the answer is everything I say is important and can be on the test, but in reality, you're not going to be memorizing any structures. You're not going to be drawing this to show me that you know what the structure is. On a multiple choice test, what might you be doing? Well, you'd need to recognize reasonable mechanistic steps. You would need to recognize which arrows form which bonds. And you would need to be able to explain some of the regioselectivity where possible. For example, why does carbon 4 form a bond with carbon 2 instead of a lot of other possibilities? Okay? All right. And the other reason we did this is just to show you how reactions that really you learned about in 351 prepared you entirely for all of the chemical steps here. Alkene addition, carbocation stability, epoxide opening, all of that's 351 stuff, but now you understand the chemistry or you understand at the level of what bonds are forming and the rearrangements, how we get from a simple linear precursor to linosterol. And I want you to see how cool it is, well, cool is a relative term, that along the pathway, every time there's an electrophile, there's an alkene either 5 or 6 atoms away that can do the attacking to close up a ring. And that is governed by the terpene structure that's present even in these very simple starting materials. Okay, so that's it for cholesterol. But that's not all, because we're going to now move on to chapter 26, which is about nucleic acids. So we're going to start by talking about the 4 or 5 heterocyclic bases that are important in the hydrogen bonding of one strand of DNA with another. And the identity of these bases is important because their identity forms the basis of DNA replication, mRNA transcription, and then protein synthesis. So there are 2 kinds of what your text calls heterocyclic bases. They're called heterocycles because they have heteroatoms in the rings. They're called bases because, as you will see, they have lots of nitrogens that have lone pairs in sp2 hybridized orbitals that you would expect to be basic. There are 2 classes of heterocyclic bases. There are the purines and the pyrimidines. The pyrimidines are, and you will encounter that. I'm not going to ask you to memorize that term. Pyrimidines are simply 6-membered rings. Purines are fused 5 plus 6-membered rings that share one side. So we'll start with adenine. And the core structure of the purine is the same in both adenine and guanine. And what I mean by that is where the nitrogens are and where the carbon sigma bonds are. So this is the basic sigma framework of a purine. In adenine and in guanine, we're going to have double bonds there. The lone pair here is not part of the pi system. It's in an sp2 orbital. This nitrogen is, if adenine were just on its own, would be bonded to a hydrogen, and this lone pair is part of the pi system. However, in DNA and RNA, this nitrogen is going to be bonded to the anomeric carbon of ribose. So I'll just draw a line with the squiggly line crossing it to indicate that there's ribose down here. On the other 6-membered ring, you've got double bonds here and here. These nitrogens have lone pairs in an sp2 orbital, so they're basic. And then you've got an amino group outside the ring, and this is sometimes called an exocyclic amino group because it's outside the ring. Now, a couple other interesting things that we're going to keep track of here. As we talk about these bases, we're going to use, let's just use red. Let's use red for something that is a hydrogen bond donor. By this we mean an electron-poor hydrogen. And then we'll use blue, oops, that's not blue. We'll use blue for a hydrogen bond acceptor, which is going to be a nucleophilic lone pair, basically. So on adenine, you have a hydrogen bond donor in these NH bonds, and then you have 2 hydrogen bond acceptors, the nitrogens with lone pairs. And actually, you have another one here. And we're going to see that this part of the molecule is what we're going to call the Watson-Crick face of the molecule. This is the part that can participate in Watson-Crick hydrogen bonding in double-stranded DNA. All right, questions about adenine? I will ask you to memorize the structures of these bases. As I said, if you get it wrong on an exam, you can challenge me to draw it in my office, and if I get it wrong, you get points back. And to avoid individual and repetitive challenges again and again and again, I will say if I get it wrong, everybody gets points back. Is that better? Okay. Okay. So, all right. So next we'll do guanine. And guanine has the same basic ring structure as adenine. You've got these two nitrogens in the 5-membered ring bonded this way. The 6-membered ring in guanine is a little bit different, not in the placement of the nitrogens, but as to whether or not they're participating in double bonds. Let's see. Okay, so in guanine, this nitrogen is an NH group. You've got a carbonyl oxygen here, and then you have an exocyclic amino group here. Double bond there. Guanine is still aromatic. The lone pairs here can form by resonance a pi bond between this nitrogen and that carbon. And so if a molecule is aromatic in one resonance structure, it's generally considered aromatic in all of them. I'm not going to ask you whether adenine or guanine is aromatic because it has an easy answer, and the answer is yes. So, what would be the point? Interestingly, guanine has more groups that are either hydrogen bond donors or acceptors than does adenine. So you've got a donor here. I'm sorry, I'm getting that wrong. This is in blue are the hydrogen bond acceptors, and then in red are the hydrogen bond donors. The reason I get that wrong is because I'm used to thinking of donors as nucleophiles and acceptors as electrophiles, but the language for hydrogen bond donating and accepting is backwards from that. All right, those are the purines, and notice for guanine you've got acceptor, donor, and donor. Whereas with adenine, there was no additional group at that carbon. All right, now let's do the pyrimidines. These are 6-membered rings, and the first one we'll talk about is thymine. And thymine looks like this. So you've got a nitrogen, and that's the one that's bonded to the anomeric carbon of ribose. You've got an NH group here. You've got a carbonyl oxygen here and here. You've got a double bond there and a methyl group there. That's thymine. You've got acceptor, acceptor, and donor. And uracil is just no methyl group. And uracil is found in RNA, whereas thymine is found in DNA. All right. And we're going to see that this oxygen here, the donor here, is going to be complementary to the exocyclic amino group acceptor there, whereas this NH bond donor is going to be complementary to this ring nitrogen acceptor. Interestingly, this carbonyl isn't going to have a similar acceptor or donor partner at this portion of adenine. That will stick out into either the major or the minor groove. I can't remember which. And then lastly, we'll draw cytosine, which is going to have the same pattern of nitrogens, but a different pattern of donors and acceptors. Cytosine has an exocyclic amino group at the top, and then it has two other hydrogen bond acceptors, the ring nitrogen and a carbonyl oxygen. And notice the complementarity here. In guanine, you have acceptor, donor, donor. In cytosine, you have acceptor, donor, 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 acceptor, acceptor. At some point, I'm going to get that right, but probably not soon. Okay, so what I want you to do for homework before next time is I want you to draw a hydrogen-bound pair of adenine with thymine, and I want you to draw a hydrogen bond pair of guanine with cytosine. All right, that'll do it for today. Have a great day.